Okay, so it's 11. Let's start again. I'm very happy to have Dr. Jessica Abbott here with us today. Jessica is a senior university lecturer at the University of Lund in Sweden. And she's a member of the DROS-EU network, the Drosophila Population Genomics Consortium. And she has kindly agreed to explain to us a little bit of her research. And she's gonna tell us, she's gonna share with us why we should care about fruit flies. Jessica, the floor is yours. Thank you so much for being here.
Okay, thank you for inviting me. I'm actually in Spain at the moment, visiting the University of Valencia. So I'm sitting in a seminar room and there's some sort of machinery going on outside. So if the noise is distracting, then let me know and I can try to put on the noise canceling function on my headset here.
Okay,
so we'll see. I'm not sure, it might not be picked up by the microphone though. All right, I'll share my screen. Okay, so I picked the title of my talk because this is something that I often get asked by people is, you know, why should I care about fruit flies? What's so interesting about them? Let's see, screen sharing is paused. Can you guys see the?
No, yeah, we'll see you.
Okay, good. Just thought I'd double check.
Wait.
Yeah, and it's moved forward now as well.
No, we see on gray. Let's see what's going on.
Okay.
It has disappeared. You were right, Jessica. Yeah.
Yep.
We could see it, but then it stopped.
It said that it was paused, so I'll try again.
Mm-hmm.
Can you see the experimental evolution slide?
Yes.
Okay, perfect. Yeah, so I thought I'd tell you a little bit about what I do with fruit flies, or one of the projects that we've worked on recently. We do many different studies, and so I can only talk about something kind of short in such a short time. But one of the main methods that we use in my group is experimental evolution. And the way this works is that you have some sort of experimental treatment that you apply to the flies. It could be a difference in temperature, for example. And then it doesn't want to move forward. There we go. And then you let a number of generations pass. So you let them live in this changed environment for a while. And then after time has passed, then you can look at the flies and you can say, okay, how have they changed? Have they changed? And is it in the way that we expected? And then the genomics part is that you can then look and see what genes might be underlying these changes. And one of the things that I've been really interested in recently is to do with the evolution of sex chromosomes. And in that sense, flies are really useful because they have XY sex determination the way humans do. So as you probably know, men, they have X and Y chromosomes and women have two X's. And it's the same with flies. The males have an X and a Y and the females have two X's. So we've done various experiments trying to look at sex chromosome evolution in this species. And just a little bit more background before I start talking about what we actually did. So the main sort of theory about how sex chromosomes evolve is that you start off with a regular autosome, chromosome that's not special in any particular way, and you get a sex determining gene that arises on this autosome. And so then you might get sort of, on different chromosomes or in approximately the same place, you could get a female version and a male version. So you get some sort of feminizing mutation and some sort of masculinizing mutation. When this happens, then you would expect that you should get a recombination stopping. So this genetic exchange between homologous chromosomes should stop so that you can keep male benefit genes together with the male determining locus, and you can keep female benefit genes with the female determining locus. And this is how sex chromosomes then specialize over time to be sort of more adapted for male or female fitness. And as this process goes on, without recombination, then you can't repair mutations to the DNA in the same way, and the Y chromosome tends to degenerate and it loses genes and it often gets smaller. So one of the things that we wanted to look at a few years ago was if there's a possibility of coevolution between the X and the Y chromosome. So can it be the case that, if you, for example, have a mutation that arises on the Y, then it could fix just by chance. So it could spread throughout the population by chance. And then all of the males that have this Y chromosome would maybe be doing slightly worse than if they didn't have it. So where our idea was that then the X chromosome might be able to compensate for this somehow. So if there was a mutation on the X that could compensate for any sort of bad thing happening on the Y, then you would reestablish normality, and then that would be a good thing. But because what happens on the Y is probably going to be different in different populations, then we should have different combinations of slightly bad Y mutations and compensating X mutations. So that was what we wanted to look at, was to test this idea that you should have different types of compensating mutations in different populations, so that if you took an X and a Y from a different population and put them together, then the males should do worse. They should probably not be very fit. They should be, not very happy. So we decided to test this experimentally. This is a super complicated figure, so I'm going to show a more simplified version. But we brought together X's and Y's from different populations and used the special crossing scheme. So we had 5 different populations from different parts of the world. And we wanted to see if we could see any effect of males with mismatched X and Y's being not very good. So the sort of simplified version of this big crossing scheme, if we just take 2 populations, in this case, a yellow and a green one, then in the control, we would have the green population with all of the chromosomes matching and the yellow population with all of them matching. But then we would have either a novel X sort of treatment where you have a mismatched X and Y, but the Y matches with the rest of the genome, and a novel Y treatment with mismatched X and Y, but the X matches with the rest of the genome. And then doing this, we wanted to see if we could sort of disentangle effects of interactions with the other chromosomes. And we expected, as I said, that males that had these mismatched chromosomes should have fewer offspring. So we tried this, and we found something totally unexpected, which was that it was the opposite. The males that had the mismatched sex chromosomes actually had higher fitness. They were doing a little bit better. So this was very weird. We didn't really know what to think about this because it wasn't what we thought we would get. So we decided to follow up, and so we thought, okay, well, is it something to do with the X and the Y mismatching, or is it the fact that it's the X and the Y is mismatching with the rest of the chromosomes? So what we did then is we created flies. We crossed an X and Y that matched into an autosomal background that was different, which we called this novel XY then. And there was no difference. So obviously it wasn't something to do with the interactions with the autosomes per se. It was more something to do with the way the X and the Y interact with each other. We tried to measure a bunch of different things. If this type of mutation on the Y, that when it's mismatched, actually is good for males, then why don't all of the populations have it? Well, there must be some cost associated with it, we thought. So we looked to see, number one, was the effect stable over time? And no, it disappeared. So after 25 generations, there was no difference anymore between the males with the mismatched chromosomes and the ones that had the matching chromosomes. So this, again, sort of strengthened our suspicion that there might be some sort of cost, that this might be bad somehow, either to the males themselves or to the females. And we measured a bunch of different things. And the only thing that we really were able to find much of a difference was offspring survival, particularly the ones with the novel X. They seem to survive a little bit less well. But we still aren't 100% sure if we've captured what the cost was in this case. But yeah. That's the working hypothesis. So we developed a new idea from this, actually, that this Y-linked mutation that we assume is the start of this process is probably good for males somehow, but bad for females. So maybe it makes the males do better in sperm competition, but is harmful to their mates, for example. Although we didn't find any direct example of that, but somehow affects the females so that the offspring survive less well. And that's bad for the mates of these males, obviously. This would mean that any female that has some sort of mutation that would suppress the effect of this Y should be favored, and that you would restore the balance between the sexes again. And so then we think that this is what's happened, and that's what we think has happened actually when we let these populations with mismatched sex chromosomes exist over time, that maybe this somehow is selecting for variants on the X that were already there, but that could suppress whatever Y-linked mutation we had. And so again, then we would think that different combinations would arise in different populations, and that this could then be related to speciation. So there's a pattern that we see in a lot of species that if you hybridize between two closely related species, then it's often the sex that has two different sex chromosomes that has worse survival or worse fertility. So if you cross a tiger and a lion together to produce a liger, then the females can be fertile, but the males basically never are. And there's lots of examples of this in birds. Birds have the opposite sort of sex chromosome system where the females have two different sex chromosomes and the males have two of the same. It's called ZW system. And there's many bird species where you can see this, where it's actually the females that have poor survival or are infertile. So we think that this could be contributing to this process of differences between species that's related to the sex chromosomes. We also developed a mathematical model to support our hypothesis, and I won't go into the details of the model because it's kind of complicated, but it basically suggested that yes, we don't know 100% for sure that this is what was happening in our experiment, but it's plausible at least. The mathematical model suggested that yes, if you do have a mutation that arises on the Y that's good for males but somehow bad for females, then the X chromosome is more likely to have a suppressor mutation arise. It's more efficient as a suppressor, and it's more likely to go through a sort of cycle of, you know, the Y-linked mutation becomes common and then the X-linked mutation becomes common. And then you might have a new Y-linked mutation at some point and then a new X-linked mutation. So that was really exciting, I think, for us to come up with basically a new theory from some unexpected results. And I know that when we got these results, this was supposed to be a kind of quick and easy experiment, you know, and it ended up taking years to complete because we had to spend all of this time trying different hypotheses and, you know, developing the model and all the rest of it. But I'm happy with the way that it worked out, because this really demonstrates how the scientific process can work, that you might have an idea, it turns out that you don't get the results that you expected, and then you have to, you know, sort of move the field forward by trying to find new information and new hypotheses. So getting back to what I said in the beginning, why should we care about fruit flies? Well, I like them because they're a great model organism. They're really easy to work with in the lab. They have this XY sex chromosome system, which is not of the same origin as ours. The genes are not homologous, the chromosomes are not homologous to us, but the system is. And so then you can say things about selection on the X and the Y that still could be, you know, relevant for other species. And they can help us understand everything from human diseases to, as I was saying, speciation processes. So they're really useful for, I think, studying these underlying processes and theories so that we can then go and see if we can see similar things happening in other species. And one of the reasons why I think it's interesting to work on sex chromosomes in fruit flies is because, as I was touching on just now, studying other species can help us understand humans. So by understanding what causes sex chromosomes to evolve and sex differences to evolve in other species, then that can say something about human sex differences. Why are we similar in some ways and different in others? Okay, so that was basically all I wanted to say. I wanted to try to leave a little bit of time for questions, and it looks like I managed, so thank you. And I'll stop sharing.
Okay.
Yeah. Thank you so much, Jessica. It was a perfect presentation, as you mentioned, no, of how science works. No, how often we have a working hypothesis, we do the experiments, expecting to get some results, but we get the opposite ones.
Exactly.
And this is often difficult for the students. I have experienced this at least. But this is really how science works. So these unexpected results are the ones that very often lead us to new discoveries, right? So we have to think again and create a new theory and do more experiments. So thank you. It was beautiful and a perfect talk, I think, for this audience.
Oh, good. I'm glad to hear you say that. And well, you're exactly right. The person who was working on this project was a PhD student, and she hated it. She thought it was so frustrating that she wasn't getting the results that she was supposed to get, and that it was, you know, taking all of this time, and that it was really confusing. And the whole time I was trying to pepper her. I was like, it's good, it's a good thing, it's exciting. But in the end, you know, when we finally were done with everything and got it published, and, you know, we're able to come up with something new, then she changed her mind. Then she agreed.
but this is, I think it's very, very important, no? Because often students just want to with the first experiment, they want to get the answer that they were expecting, and this is not how science works most of the time. So wonderful. So if there is any question for Jessica, anyone, you can write them down in the chat if you prefer, or you can open the microphone. Si hay alguna pregunta para Jessica, si la queréis hacer en castellano, la podéis escribir en el chat, si queréis, o la podéis decir y yo la traduzco al inglés.
Yo tengo una pregunta para Jessica, mientras.
Muy bien.
Jessica, in the context of your research and draws you in this different context, yourself draws you in citizen science, how in this project, how do you see what are the perspectives or the potential that you see, or your thoughts about citizen science, for example?
Yeah, so in this case, we've had students, high school students, coming into the lab and helping out with some aspects of our projects. And so, yeah, I think we maybe have had a little bit of help with this one. But so I participate in a program in Sweden where they give scholarships to high school students to work in our lab for 2 weeks in the summer. And so it's normally 2 students a year that we take in, and then they can help do pilot projects. They can, you know, help collect data and just help out with things in the lab. So that's kind of citizen science on the small scale. So when we're doing stuff in the lab, then, you know, it's hard to get people involved on the big scale. But that's what I think is really exciting about the DROS EU initiative, is that, you know, being able to do these collections and get lots of flies from different places so that we can get much more data than we would be able to do without the help of all of these, you know, students and citizen scientists. I think it's great. It's really, I think that popular science, you know, communication is a really important thing. I try to spend time doing it, and so I think that this is a super initiative.
Thank you. So, okay, you're welcome. You know, you have this entire network to use to your disposal. So,
exactly.
Any suggestion you can share with us? We have this amazing team.
One of the things that I was thinking about, because I have done field work, you know, during my PhD and stuff, but since then I've been mainly doing stuff in the lab. But I was thinking that it's also nice to be part of this network so that you can, you know, meet people who are experts in other different things. So if I wanted to learn about viruses in flies, or if I wanted to, you know, study predation or something, then there are people who are adaptation to temperature, then there are all these people who know about this, and then we can work together. And that's also really helpful for moving science forward, you know, creating this network of people who can help each other out with different questions and things.
Okay, we have one question from the ES La Correduría.
Yes, one moment, please. Hi, we wanted to ask how long took you, Jessica, to develop the whole experiment you did?
Yeah, that's a good question. So I think we did the experiment first in maybe 2014 or something, with the cross. So to cross the X and the Y from different populations together, we used a special set of flies that have, you know, special chromosomes that mean that you can control the inheritance of all the chromosomes. So to set up the populations took like 5 generations of crossing, I think. So that in itself was a little bit of time. And then we did the experiment, and then we spent a year or so afterwards trying to do other experiments to follow up on this and see how they differed and do this experimental evolution part. So we did all of that, and then we spent some time analyzing the data, and then we realized that the experimental evolution part, we'd used a method that maybe wasn't optimal. So then we did everything again, basically. We did all of the crossing and measured the fitness again, and then did the experimental evolution part again, and then that took about another year. And then we had all of the data and we wanted to develop this model. So then we waited for a little while until I was able to hire someone who was a mathematical modeler who could work on that. And then he spent about another year working on it. So it wasn't working on it the whole time. We had other stuff going on at the same time, but it was a very long process, actually. So the experiments themselves took about 2 years, but then there was a bunch of other sort of writing and analysis and developing the model and just sort of waiting for the right time to do the next part. Yeah, so it took quite a while.
Yeah. So we have another question, and they are asking if there is any relationship, or what is the relationship between what you explain about sexual chromosome evolution and speciation through segregation distorted genes.
Yeah, and that's a good point, because these segregation disorders are expected to be associated with sex chromosomes often. And what you normally think, or what people normally expect, is that anything that is causing an overproduction of males is doomed to disappear quite quickly, because the population will just collapse. So you get more disorders that will be X-linked. And then you can expect that you will have interactions between sort of these X-linked disorders and then autosomal or Y-linked suppressors of various types, and that can lead to a similar sort of process. So we talked about that in the paper, actually, that it could be related to these meiotic disorders, but we measured the sex ratio in the offspring and we didn't see any bias. So it didn't seem like that was what was going on in this case, but it certainly can be.
Thank you. Any other question? Anyone? Either through the chat or live? Okay. If there are no more questions, once again, thank you so much, Jessica. It was great to have you.
Yeah, no problem. Thank you for inviting me.
Thank you. And looking forward to see you again.
Yes. Everybody enjoy the rest of the day.
Thank you.
Okay.
Bye.
Thank you.
Bye.
Bye. Take care.
Okay, so I think we can now move to Christian Feller and his students. I'm seeing that he's already sharing his screen. That's wonderful. And they are gonna explain us evidence for a new transposable element, jumping gene, in 2 different Drosophila melanogaster populations. So Christian and students, the floor is yours. We can see the presentation. It's in presenter mode. We cannot hear you yet.
Can you hear us?
No. Yes,
now we can hear you.
Perfect.
But it's in presenter mode now.
Yeah.
Yes, it should be.
Okay.
Okay, perfect. Go ahead, please. Thank you.
Welcome.
All right. So I am Manuel. And as you can see, we are here to present our discovery, if you can call it that way, of the new transposable element into Drosophila melanogaster populations. Before we do that, though, we want to quickly introduce ourselves. We are from the Justus von Liebig School in Germany, Waldshut. And we were originally 23 plus students who worked on all the experiments, which we're going to talk about in a bit. The 8 that you can see on this picture helped writing the research report. And the 4 of us are going to present our results. And of course, we have to mention that we could have not done this without the support of our teacher, Mr. Feller. Yeah, I think we can continue now.
First of all, student partners group, like some of you, collected, classified, and sent fruit flies to the researchers. The researchers then analyzed the flies' genomes and predicted novel TE. And that's where we come into play. Our job was to test the predictions for 5 novel TEs in the fruit flies populations.
And that was basically our main goal, to test these predictions. But of course, in more of a personal level, we wanted to work with researchers and acquire knowledge that we could use for future projects in the field of biotechnology.
Now we have mentioned the word transposable elements quite often, but what are they exactly? Transposable elements, or transposon, or as we refer to the rest of the presentation, these are DNA sequences that can change their position. This can happen in different ways, but here we are going to show you a simplified version of the most basic process. Here, you see a so-called donor DNA, which contains the TE. Under particular circumstances, the TE can excise itself from the donor DNA, and when it meets another DNA strand, which we call a target DNA, it can integrate itself into the targeted DNA. Think of it as a sort of cut and paste mechanism. There are many reasons as to why scientists are keen to discover new TEs. First of all, they increase genetic diversity, and they are considered to play an important role in evolution and adaptation. For example, climate change. However, they are also not the easiest to find. On one side, they are highly repetitive, and they are not presented in an individual of a single species, but they are quite rare.
Now that we have talked about the importance of TEs and the problem of finding them, let's move on to our work. To remind you, before we did our part of the project, the researchers analyzed the flies' genomes and predicted novel TEs. Now to our work. Firstly, we retrieved flanking DNA regions to the predicted integration site. After that, with NCBI primer-wise, we designed 2 primer pairs for each predicted. Then we homogenized adult male fruit flies and extracted DNA. When that was completed, we performed over 100 PCRs. In this picture, you can see a PCR machine. Last but not least, we used agile electrophoresis to see which fly population exhibited transposable elements.
What is gel electrophoresis? I'm going to explain a simplified version of what happens during gel electrophoresis to you. What you can already see on this slide is an electrophoresis gel. It has a net structure, and at the top, you can see pockets in the gel in which the DNA is going to be placed. As an example, a DNA molecule with a length of 400 base pairs. The gel is an electrically charged field, and since the DNA is negatively charged, it moves downwards to the negative charge, and the DNA molecule creates a band at a specific length, which you can see under UV light. In this example, a band is shown at around 400 base pairs.
And what is it going to look like if we have a DNA molecule with an integrated TE? First of all, the PCR product is going to be longer, and instead of 400 base pairs, it might be 1000 base pairs long due to a TE with a length of 600 base pairs. If we put that on the gel as well, we are going to see a band too, but it is going to be not as far down as the first one. So if we get a band that is higher than we expected, that is shifted upwards, then there is most likely a TE within it.
Now let's take a quick look at the results we got. On the gels, you can already see 2 DNA size markers, which help us declare how large a DNA molecule is. Shorter DNA runs faster, as we can see here by the band of a DNA molecule with a length of 200 base pairs. Longer DNA needs more times, but a DNA molecule of a size of 1000 base pairs is up here. On the bottom, you can see 5 different fly populations that we tested for TEs, and we tested each possible placement of the TEs by 2 different primer pairs, which makes our results more confident. As a first result on this primer pair, you can see for F1 and F2, each a single band around 200 base pairs, and from the placement of our primer pair, we can see no TEs and therefore no shift of the band. And however, if you take a look at fly population 3 and 5, you can see a higher molecular size, so the DNA fragment jumps from 200 base pairs to 1000 base pairs. And we can see a shift. Both primers for F5 generated a shift of approximately 800 base pairs in the PCR product. But really surprisingly, only this primer, TE32, generated a jump of the DNA fragment in F3, while this primer over here, TE31, generated a PCR product of the expected size. So all in all, our results suggest the existence of TEs as predicted by the bioinformatic analysis in the flight population F5, which comes from Raleigh, North Carolina, USA, and surprisingly also in F3, which comes from Munich, Germany, although this was not predicted. This means we found a new TE.
The bioinformatical analysis regarding the position of the flight genome. We found out that TE3 is located at the center of the X chromosome to these two positions. And TE3 is located with the first large intron of this gene and 2 kilobases downstream of that gene. But what's really interesting about that is that almost nothing is known about these two genes. That's why they don't have actual names.
Yeah, before we come to your questions, of course, we want to give a big thank you to Dr. Miriam Merenciano and Professor Dr. Josefa González for initiating this project and giving us the opportunity to take part in it. And yes, thank you all for listening. If you have any questions, we will gladly answer them.
Thank you for participating. And the presentation was very clear. And your gels, your electrophoresis gels are just beautiful. So congratulations, very clear ones. Yeah.
Thank you.
So if anyone has any question for the students, again, you can write them in the chat in Spanish or in English, or you can connect your microphones. Si tenéis alguna pregunta, igual que en la charla anterior, la podéis escribir en el chat y yo la traduzco al inglés, o la podéis hacer en directo.
I have a question for them.
Okay, go ahead.
After this experience with the Citizen Fly Lab, what's next for you? What are your perspectives? Are you encouraged to do some new experiments? What are you planning? What are you thinking about something?
Do you mean on a more private level, or like what we want to do with, because we did this experiment in school, you know?
Yeah, any level.
Right now, because we just finished this, I don't think we have anything as big planned for the foreseeable future, but of course we would like to work with other researchers and do other similar projects.
And actually, we and my friend over here, we have a project plan for a German science competition where we're going to look at the TEs more closely and TEs from all over the world, and if there is a connection between climate. If one climate there's the same TE as in another climate, but yeah, that's for the future. So maybe next year we can present that. We don't know.
We're looking for that. Looking forward for that. Any comments, any thought of the overall experience, personally, professionally, academically? What has to represent this experience for you?
I would say this definitely represents kind of a starting point for us in working in this field of science. And like I said at the beginning, we hope that we can do more similar projects. And yeah, we see this as a solid base where to start. We did all the experiments, and now we are pretty confident in the way that we perform them, and we hope we can do more.
Okay. So we can say we have new Drosophila fans here.
You can say so, yeah.
Okay. I hope you became Drosophila influencer as well. Okay. Thanks so much for your work, really.
Yeah, thank you.
You're welcome.
So if there is no, you are getting some clapping from the other schools.
Thank you.
I have to say, I love your graphics. As a graphic designer, I love the graphics of your presentations.
Okay, great. So if there are no other questions or comments, I think we can continue. We can move forward to the next talk, which is going to be by Dr. Iwan Harney. Iwan, oh yeah, he's ready.
You can hear me?
Oh yeah, yep, we can hear you. So Iwan is a postdoctoral researcher in Barcelona in our lab, and he's gonna explain us about how do wild populations of Drosophila melanogaster respond to heat stress. Iwan, thank you so much for agreeing to give this talk, and the floor is yours. The virtual floor is yours.
Okay, thank you. Let me just share the screen. Okay, can everybody see that?
Yeah.
Yes.
Perfect, thank you.
Great. Okay, hello everybody. Hola a todas y a todos. As Pepe said, my name is Iwan Harney and I'm a postdoctoral researcher working in her lab. And I'm studying how wild populations of Drosophila melanogaster might be responding to heat stress. And before I start, I'm just going to kind of wonder if any of you might have seen these kind of colored stripes on the left here, these blue and red colored stripes. They're known as the warming stripes, and what they do is they show how changes in global temperature have changed between 1850 and the present day, compared to an average determined from the 1970s to the 1990s. And I think they do a really good job of showing how global temperatures have increased by more than 1 degree over that time period. So clearly, global climate change is a problem, and due to greenhouse gas emissions from human activity, we're seeing the world becoming a warmer place. So this doesn't only mean that the ice is melting in the poles, but also we're seeing more extreme events like hurricanes, wildfires, floods, and droughts. Clearly, this is going to create lots of problems and challenges for human society, but it's also a problem for many organisms as well. And as biologists, we're really interested in trying to understand how they're going to respond to these challenges and how they might adapt to them. But the question is, how do we do this? So as well as studying, one important way of doing this is to test how organisms do respond when they experience variation. So when we have variation in climatic factors like temperature and humidity, we know that humans respond, for example, to cold by having goosebumps, and when we're hot, we get sweaty. But for many organisms, they have different responses, and it's important to try and understand the physiological and behavioral changes that might be caused by changes in the global climate. It's also important to compare populations that have experienced different conditions. So for example, here we see different climatic conditions across Europe, and we might expect that an organism from Spain or from Portugal, from the Iberian Peninsula, will have very different adaptations to one from Northern Europe, from the Nordic countries. So as an example, the mountain hare, which we find in Northern Europe, has white coat during the winter to help it camouflage in the snow, and it has small ears, which help it retain its heat in cold conditions. On the other hand, the Iberian hare has large ears so that it can lose heat because it gets hot. These adaptations took millions of years to evolve, however, so these are very different species. Global climate change, on the other hand, is happening over a much shorter time period. So to understand these adaptations over a short time period, we need to find the right model organism to answer these questions. So the ideal organism should have a wide distribution so that we can still study things like the warm Iberian Peninsula and cold Nordic countries. It should also have a quick generation time so that we can study multiple generations in the lab. And for no surprise for anyone probably, I think that the fruit fly is a fantastic organism that fills these requirements. And of course, everybody here knows quite a bit about Drosophila and are quite familiar with the two species that are common in Europe, Drosophila melanogaster and Drosophila simulans. For me, I'm particularly interested in Melanogaster because we know so much about its genetics and genomics, and as we saw very nicely in the last couple of presentations, it's really a fantastic model organism from the genomic perspective, and I will mention a little bit about that at the end. But moving on, what I want to talk about today is what I've been able to do with some of the catch the fly samples, which have been sent to me by all the different schools from across Spain, and which I'm very grateful for. So thanks to Catch the Fly, I've been able to study flies from sent by 12 different schools from across 7 regions, as well as a site sampled by the University of Barcelona in Gimenells. And all the schools sent me live flies. They separated out the males from the females and just sent me the females because I need the females to continue the generations in the lab. Once I received the flies, I double-checked to make sure that what I had were females, and if there were any males left in there, I got rid of them. And then I put the females in individual tubes. So the reason why I had to keep the females in individual tubes is because, as you all know, it's quite difficult to tell Drosophila, Melanogaster, and simulans apart by the females. And actually, it's the males that we use to distinguish the 2 species. So in order to distinguish my flies that I received from all the schools, I kept the females individually, and I waited for her offspring to appear, and I used her male offspring to then figure out whether or not she was Drosophila simulans or Drosophila melanogaster. So I'm going to go on to the data in a second. I also need to apologize to one of the schools on this map, which is the Colegio María de Olivar Según in Valencia, because unfortunately they sent me lots of flies, but I received them too late to include in the experiment. So I'll be presenting from the other schools, but not them, unfortunately. So here is the data about males and females in terms of the percentage of females to males. The bars go from north to south, so the bars on the right kind of correspond with the locations on the map. And as we can see, the females in purple, we have a majority of females, which is great, of course. There were some males in there, but that's not unexpected because it's quite hard to distinguish the males from the females and do the sorting when the flies are alive and flying around and trying to escape. And if we move on to the percentage of the 2 different species, Melanogaster and simulans, here Melanogaster are in red and simulans are in blue. We also have discarded flies in grey. So these were flies that females which either they didn't lay any eggs or they laid eggs and they were only females. So I wasn't able to tell which species they were. And looking at this data, we can see that for all the sites, actually, the majority of flies were Drosophila simulans. However, there were some sites, in quite a few sites, we got small numbers of Melanogaster. And in a few sites, we got reasonable numbers of Melanogaster. So these were really the ones that I wanted to focus on for my following experiments. And the schools that sent me these samples were Colegio Niño Jesus in Burgos, the ES Benjamin de Tudela in Tudela, and the ES Atuer in Manzanares. So these 3 sites, they're not the most distant in terms of the geography and distance, but they do have different climatic conditions. This is based on weather station data from nearby cities. We can see that Burgos has lower mean average temperatures than both Manzanares and Tudela. And if we look at the daily difference in the temperature, so that's kind of the daytime maximum minus the nighttime minimum, we can see that Burgos actually has very high difference in the summer. Manzanares has very high difference in the winter, and Tudela has the least difference. So we do have some variation in the types of climate that these flies are going to experience. Okay, so as well as including our Spanish populations, we also wanted to include some populations which have maybe had a completely different climate, not just this kind of small amount of variation. So thanks to researchers from the University of Edinburgh and the in Scotland and the University of Jyväskylä in Finland, we were able to sample a couple of Northern European populations from Edinburgh and from Akka. And once I had received those flies and everything was ready to go for the experiments, I decided to focus on the Finnish flies and these 3 Spanish populations from Tudela, Burgos, and Manzanares. Just to give you a kind of overview of what type of climates they experience, Manzanares and Tudela are considered to have something between arid steppe climate and temperate dry summer, hot summer climate. Burgos is more generally temperate with less aridity and a warm summer but not a hot summer. Of course, on the other hand, the population from Finland, from Akka, has a completely different climatic regime, which is cold and humid. So these are really, really different climates that they experience. So the question is, do these flies have adaptations that might help them respond differently to these climates? And also, might they be visible might these adaptations be visible if we expose the flies to some kind of heating stress? And that heat stress is something that's likely to happen as global climate change occurs and temperatures increase. So we tested this. We have flies maybe with adaptations to the climates that they've evolved in. So how might we test this? Well, we use one of the traits we use is called CT max, and this is the critical thermal limit of a fly. For cold-blooded organisms, including flies, their performance, which is their ability to do anything really, to move, to eat, to behave normally, varies across a range of temperatures. And most organisms will have an optimum temperature, this TO. And if we put them above that temperature, they'll be comfortable at that temperature, and if we increase the temperature, they may not perform as well. So, for example, if we put our fly in a tube, we can control the environment and heat it up slowly and see what happens. And if we do this, what we're likely to see is that its performance will decline and it will become stressed. And at some point, it will reach its maximum temperature and it will fall over and faint. And this is its CT max. It hasn't died, it's just got so heat stressed that it's passed out. So in the lab, we do this by slowly heating tubes in a water bath, a baño Maria. Here's a short video of what this looks like. As you can see, there are a few tubes here, each with an individual fly, B for Burgos, A for Akka, T for Tudela. And you can see the flies moving around. And as we increase the temperature slowly, their behavior will change. But at some point, when they get too hot, they'll stop moving and they'll fall over. And at that point, I take the tubes out because we're not trying to harm the flies really. We just want to see what their critical temperature is. So I measured this CT max trait for 32 different flies from 4 different populations. And what we found is that there was a small but significant difference between the populations in their CT max, or their fainting temperature. And what we can see here is that flies from Manzanares are able to tolerate temperatures nearly half a degree higher than the flies from Akka in Finland. Tudela also has a seemingly lower temperature, and Burgos is kind of intermediate. So as well as measuring how heat stress affects adults using CT max, we can also look at other important biological traits in different experiments. And one important thing to consider is maybe how heating stress is going to affect the life cycle of the fly and the reproduction. So what we can do, for example, is measure the fecundity, the number of eggs laid, and we can also measure how many of those eggs turn into pupae, or turn into adults. And this gives us something known as the viability, which is basically the percentage of eggs that successfully emerge into pupae or successfully eclose into adults. And what we might expect is that if we heat the flies up and stress them, they might have lower viability, and obviously this is bad because they're not going to be able to pass on their genes to as many offspring for future generations. So for this experiment, I had 3 different treatments. There was a control treatment with no heat stress. There was a treatment which was exposed to 36 degrees for an hour, and a treatment that was exposed to 37 degrees for an hour. And I basically assessed the viability, the percentage of eggs to pupae at 2 different points. After 48 hours, female to lay eggs for 2 days. After 2 days, the female was transferred to another tube, again allowed to lay as many eggs as she wanted. After 4 days, the 2 flies were removed, and then we counted the eggs, and we waited for a while, and we counted the number of pupae that emerged a couple of weeks later, or a week to 2 weeks later, as the larvae developed into pupae. And here we found that at 37 degrees, there was a significant effect of a heat stress. And this is particularly clear in the flies from Acre and from Burgos. You can see here the difference in the percentage viability between the control on the left in pale blue or pale green and the 37 degrees in the darker blue and green. The effect is less clear for Manzanares and Tudela. Interestingly, when you look at what happens between 2 and 4 days, this effect kind of disappears and there's less obvious effect of the female having been heat stressed. So just to summarize these results, it seems that flies from the warmest population in Manzanares are more tolerant of heat than those from the coolest population in Acre. And heat stress seems to reduce egg viability, especially in the flies from Acre and also from Burgos. But it does lead to some more questions. One is whether or not these effects might be present in later generations. So if we heat the females, does it affect her daughters and her granddaughters and her great-granddaughters? And we also want to know something about the molecular mechanisms that might control tolerance of heat. So I'm not going to really talk about this very much. And you've had a few talks today where people have talked about genetics and genomics. I hopefully that's explained a little bit about why we use Drosophila melanogaster as a genetic model organism. But I'll be looking at, for these populations, these 4 populations, for genomics. So that's kind of the code of A's, G's, C's, and T's that gives us the blueprint or the plan for that organism. The transcriptomics, which are the specific bits of DNA that are transcribed, which are read in order to produce proteins, especially in response to, for example, a heat stress. And also the epigenomics, which are telling us about the chemical modifications to the DNA that control how it's packed up into the chromosome, and that tells us which genes are being switched on and off. And that is where I'm going to stop. I just want to thank everyone from the González Lab, the Evolution and Functional Genomics Lab, our collaborators in Uvascular, Barcelona and Edinburgh, Roberto Torres and the Ciencia Tumon, the European Commission who fund my work, and also many, many thanks to all the students and teachers who helped collect the flies, because without them this work would completely not have been possible. So I'm just extremely grateful. I'm very happy to answer questions. También estaré encantado de responder preguntas en español y puedo probar responder en castellano o si no en inglés. Gracias.
Thank you so much, Iwan. Yes, very clear presentation. Thank you. And I also want to, again, thank all the schools that send us flies so that Iwan it's able to carry out this wonderful project. Yeah, so Jose Luis is saying congratulations, excellent work. Again, if you have any questions and you want to write them down in the chat in Spanish or in English, as Iwan was mentioning, habla un poquito de español, así que puede intentar contestar las preguntas en español también, or in Spanish. So, Antonio, buen día. They are mentioning that they have a question. Okay. You want to do it live? Yeah, go ahead.
Why are there in Tomelloso more female flies than male flies?
So could you repeat the question, please?
Why are there in Tomelloso more female flies than male flies?
Why were there more female flies? Because we asked for the schools to send us just females. So hopefully I was getting as many females as possible and just a few males that had managed to sneak in. Obviously in the populations themselves, they should be about 50% females, 50% males. But for my experiment, I needed to have just the females. So we asked the schools to send us the females.
Perfect. Any other questions or comments? Christian, yeah. Please go ahead, Christian.
So thank you very much. It was a very clear and great talk.
Thank you.
Very well understandable. There are a couple of questions here from Germany, but we're all too shy to ask, so I have to say this. So the first question was, how long do these flies live? So is there a difference in lifespan? The second question was, how fit how fit is the offspring? And the third question is there a difference in the number of total offspring? In particular, because you showed that in the early, I think it was egg laying, pupa, there was a difference, and later on not. So yeah, these 3 questions.
Okay, 3 questions. So lifespan, fitness, and the number of offspring.
Yes.
So I haven't really differences between the populations, I don't know. However, I can tell you certainly about differences in the number of offspring they have. That temperature is higher than what the finished flies are generally used to in the lab. We have them at a controlled, well, we have them at the lab temperature, which is 20 to 22 degrees, which for the Spanish flies is probably quite normal and comfortable. It's almost on the warm side for the finished flies, probably. So many organisms, many cold-blooded organisms will reproduce more and develop more quickly when they're in warmer conditions. All the metabolic rates increase and go up. So it may be that with the finished flies, because they're in a unusually warm environment, they're producing more offspring more quickly than the Spanish flies, which are in an environment more similar to what they're used to. One way we could test this actually is if we looked at we could look at the survival, because maybe the finished flies, they're doing everything quickly, which means they don't survive as long. And we could look at the number of eggs they lay over their entire life. So I've just been looking at kind of eggs laid in the first 2 days, eggs laid in the 2nd to 4th day. I've also looked a bit later, eggs they lay after 10 days for a couple of days. If we count the whole eggs over the lifespan, that would give us a real indication of fitness, and that's probably an interesting thing to do. So I may well be doing that in the future as well.
Thank you.
Thank you.
Thanks, Iwan. Yeah. If there is no other question,
I have a very general question.
Roberto, yes. Okay, go ahead, please.
Yes.
You have 2 minutes.
Okay, 2 minutes. It's very quick. It's very obvious what I'm asking, but I wanna hear you from you. How the citizen science works for you in your particular project?
Well, it's worked so very well for me. I'm very, very grateful to have been involved in this. I really can't stress enough. When I wrote this project a couple of years ago, and when I wrote the project, I said I would go to 4 or 5 different locations in Spain and try and collect flies and do all the fieldwork myself. And with the pandemic, that would have been difficult, but even without the pandemic, it would have been really a load of work and very difficult, and I wouldn't necessarily have even succeeded. So the citizen science here, where we have this fantastic network of schools going out and collecting flies and sending them back to the lab, it's made my life very easy, but I'm just really grateful, and it's really fantastic to see how we have these diverse regions represented in terms of their climate, and I'm able to study these different populations and gain information and data about the flies from these populations. So for me, it's just been an absolutely fantastic to be involved in this citizen science project. I'm really, really happy.
We're really happy to hear that. And I only want to add something for all the people to show up the relevance and the importance of this network, how it's working, the work that you're doing, the schools, is so important that you have become an asset for us in science. You're become a really valuable asset, because when we're aware that E1 was moving to Barcelona with this particular project, with this particular needs, we have the ability, we have the capability to say, okay, don't worry, we can help you with that. We have a network, a working network that can work for you. So this is a huge progress in our network, that we are able to cover all these needs. We are starting not to create needs. We are solving problems, helping directly to the advance of science in Europe, international science in all the world. So I would like that all of you, to be aware that the relevance of your work, and thanks for all your work, and thanks everyone for trusting us.
Thank you. No, thank you for giving me the chance to present this today, and thanks again to all the schools for helping.
Yep. So we have a question, but we are in the break time. So I'm gonna make the question very quick.
Okay.
So it's from the people it's from the school in Manzanares, E1, which is one of the populations that you have studied. And the question is, if have any differences in size and temperature being found in different Drosophila populations comparing Northern Europe to Southern Europe?
So I have not measured size, and I really wanted to, because in the past, in the other projects I've worked on, there's always kind of interesting things going on between size and temperature. Generally, things can grow larger. Often things grow larger in colder climates, but they also develop slower. So it's kind of a trade-off between size and age. I haven't done it here. Again, as I said with the previous questions, it's one of the things I wanted to do and that I may well do with future experiments. So now I've done these experiments, but I have the populations in the lab. So I can continue to do other experiments with them. So it's certainly something that I'd be interested to do, but maybe watch this space.
Okay, thank you so much. So now, yes, we are gonna go for the break. Please be back at 10 minutes past 12. Thank you. Thanks, everyone.